Okay, folks, we're going to get started with the afternoon session. We have a couple of talks coming up, and then we're going to move into a panel discussion. Follow that with the town hall, and then we will have some announcements. And for those who are interested in going to the cheering lab, we'll organize that. I'm going to kick it off. Do that. Okay, so we're going to start right away with Oscar Zimmerman from Northern Arizona. Okay. Try and give you a 2-minute warning.
Okay. Well, thanks, Dave. Hi, everyone. I'm a grad student, and I'm going to be talking about canopy phenology in an evergreen ecosystem today. So I might be a little bit biased, but I'm okay? Okay, thanks so much. So I might be a little bit biased, but in my view, I think it's just so important to study canopy phenology in the AmeriFlux community. And that's because phenology is one of the main biological factors driving land-atmosphere fluxes at the annual time scale. For example, in deciduous forests, the dates of leaf emergence and leaf senescence exert this first-order control on annual productivity and carbon balance by determining the length of the growing season. In evergreen forests, which is what I study, canopy phenology isn't related to changes in canopy structure, but rather seasonal changes in the physiology of the existing leaves. These changes include adjustments to their photosynthetic capacity, as well as changing sizes and compositions of various pigment pools. Since leaf pigmentation shows up in the color of leaves, we can use various remote sensing tools to track canopy phenology in evergreens. These tools include time-lapse digital cameras or phenocams, as well as other various radiometric sensors, and even our own eyes. For example, in this pair of pictures captured by the phenocam in Niwot Ridge, you can see that the needles in the wintertime turn this like rustic yellowish green color. These tools were really nicely discussed in this recent review led by Zoe Pirat, which even featured the combination of near-surface remote sensing and eddy flux data at AmeriFlux sites across the US from Alaska to Florida. But one thing I noticed in this review were the lack of studies applying these tools in semi-arid and arid regions, such as the US Southwest, where evergreen conifers form the foundations of the woodlands and forests. Since these ecosystems are limited by not only temperature, but also water availability, this could result in unique patterns of canopy phenology and phenological controls over land-atmosphere fluxes. Some questions that this knowledge gap raises include: what environmental factors drive seasonal changes in canopy color or greenness in these ecosystems? To what extent do changes in greenness regulate ecosystem photosynthetic rates? And how do changes in camera-derived greenness compare with other remotely sensed indices? To address these questions, I've been collaborating at Cedar Mesa in Utah. This site is about 1,900 meters above sea level, and it experiences a cold and semi-arid climate with cold winters and hot and dry summers. The site is registered with AmeriFlux, and it's also part of Utah Flux, which is this mesoscale network of eddy flux stations across the state of Utah that my collaborators Paul and Catherine are involved in. So if you haven't heard of their project, do check out Utah Flux because they're doing great things for the state of Utah and beyond. At Cedar Mesa, we quantified canopy greenness by calculating this green chromatic coordinate index, GCC, from phenocam images. And we specifically focused on the pixels of the images corresponding to the dominant evergreens, which were Utah juniper. So that's a picture of what the site looks from the phenocam in the summertime. And that's what it looks, or this is what it looks like in the winter after a snowfall, although the snow doesn't stay for long. We calculated rates of ecosystem photosynthesis from the measured net ecosystem CO2 fluxes. And we specifically applied this moving window light response curve approach to estimate seasonal changes in ecosystem photosynthetic capacity, or AMAX. And then finally, we also measured canopy spectral reflectance using narrowband sensors and used those data to calculate the chlorophyll carotenoid index, which is a visible wavelength index that was originally developed to track evergreen canopy phenology from MODIS satellite data. So what have we found so far? Starting with an overview of my study period, we observed that air temperature exhibited really large seasonal variation at this site. The summers were hot with afternoon temperatures in the 30s, and the winters were cold with nighttime lows well below freezing. Soil moisture was recharged by both winter and summer precipitation, but it was only the winter snows and rains that reached the deeper soil layers. And in terms of variation from one year to the next, we saw that winter in 2025 was relatively drier than 2024. And finally, even though the junipers were seasonally evergreen, we found that the color of their foliage still changed throughout the year, with greener foliage in the summer and less green foliage in the winter. And on top of that, we also saw some year-to-year variation in just how green the leaves were. Specifically, in 2025, we saw that greenness was lower than the other years. Using a temperature sum or degree days model, we found that changes in canopy greenness were mostly associated with low and high temperatures. During the autumn, greenness declined with increasing chilling degree days, and the temperature response was mostly the same between the 2 years. In spring, greenness increased with accumulating growing degree days, and again, the temperature response was the same. But as it continued to warm, the trees reached a maximum value of greenness at an earlier temperature sum in 2025 than 2024. And we think that the lower values of greenness in 2025 was due to that relatively dry winter that year. And I'm currently using phenocam data from other similar sites to try and incorporate water availability into this traditionally temperature-driven phenological model. So moving on to the CO2 fluxes. Canopy greenness was only sometimes correlated with the ecosystem photosynthetic capacity. For example, during the springtime, AMAX rose along with greenness, illustrating strong phenological control on the CO2 fluxes as these evergreens woke up from winter. But that high level of correlation dropped off over the summertime. And this last summer, we even saw photosynthetic activity completely crash, even though the canopy remained green. So we hypothesized that there might have been other things limiting photosynthetic capacity, maybe water availability. So to test that hypothesis, we modeled AMAX as the product of two limiting or reducing factors, which we represented with sigmoids. Our first limiting factor was canopy phenology, which we represented with a function of the canopy greenness. And our second limiting factor was water availability, which we represented with a function of the soil volumetric water content. Our model showed that AMAX was limited by phenology during the shoulder seasons, which is represented by the green area. But soil moisture, which is represented by the blue area, became equally, if not more limiting over the summer. This relatively simple model was able to represent not only the ramp up in AMAX in spring and its ramp down in autumn, but also this sawtooth pattern in AMAX over the summertime, which was driven by periodic rainstorms that resulted in alternating wet and dry periods. And moving on to my third and final question, we found that canopy greenness calculated from the camera images was highly correlated with this chlorophyll carotenoid index, which we measured at the crown level with narrowband sensors. This high level of correlation sort of reinforces this idea that changes in canopy greenness are associated with changes in photosynthetic pigments. And also, it suggests that we can use indices like CCI, which were originally developed to be calculated from MODIS data, to scale up canopy phenology across the broader landscape. So to summarize and conclude, temperature explained most, but not all, seasonal changes in greenness, and water availability is probably important too. Canopy greenness sets only an upper bound on photosynthetic capacity, which is further limited by soil water content. And finally, camera-derived greenness goes hand in hand with visible wavelength indices like CCI. Our hope is that analyses like these that combine eddy flux with remote sensing data will be able to help us better predict how these ecosystems are impacted by a changing climate through shifts in canopy phenology. So thanks for your attention.
Thanks, Oscar. Time for one quick question if Jen comes down. Yeah.
Thanks. This was great. I'm curious if you know off the top of your head, like what is the average just interannual variance in the summertime the greenness metric that you're using for these ecosystems? Because it's hard to tell for me if these variations during the summertime are really, really big, even though they don't look big compared to the variations in photosynthesis, or whether they're not so bad.
This site only has 2 years of data, but at similar sites, such as Marcy's Pinyon Juniper site in New Mexico, I would say there can be years where the difference is maybe 1.5 to 2 times. Yeah.
Yeah, cool.
Thanks.
Thanks, Oscar.
Thanks, Dave.
So our next speaker is Chen Wang from Indiana University.
There you go. Thanks. Thank you. So today we are going to focus on Midwest croplands. So the Midwest croplands has large potential of the carbon sequestration if we're thinking about we have been intense cultivation of the agriculture for so long time. So we actually release a lot of soil carbon from the soil. So there's a lot of agriculture conservation practices. You can actually improve the soil carbon. Thank you. Sorry. So one way to do that is cover crops. So the cover crops is the vegetation you can actually implement during the dominant season. During the Midwest is usually from the November to April. So they usually grow some high biomass grass. Those biomass hopefully could transfer to the soil carbon later on. However, the accurately quantify high resolution soil carbon is really difficult, especially at the regional scale. So there's many reasons about that. So I'm just list the reasons that I'm pretty familiar about it. So one reason is at the field level, if you're using some techniques to measure the soil core, it's usually labor intensive and also limited space and temporal coverage, but also it's also have the problems with caption spatial heterogeneity, right? So when you're thinking about the spatial scale, you might also thinking about maybe remote sensing data can help to detect something of the under the ground, but maybe some new technology is emerging, but current remote sensing data is still really good at observing dynamics above ground. So it still have like challenges really capturing anything under the ground. So directly capturing soil organic carbon is still really challenging to use some of the remote sensing techniques. So at the same, meanwhile, I think we also have this ethical variance measure the net carbon ecosystem exchange. So here I just show a really simple illustration about carbon dynamics at cropland site. So generally you take the CO2 in the ecosystem, the carbon will go to the harvest carbon, which is the yield, and then some carbon go to the runoff, and the leftover carbon might be go to the soil, right? So if we're thinking about this really simple calculations, we can actually kind of calculate the soil carbon as net ecosystem carbon balance. So the flux of the SOC might be equals NEE minus harvest carbon and minus runoff carbon. So the harvest carbon actually can be easily estimated if you just think, if you know the gram carbon content. So the runoff carbon is small compared to like NE data. So we do have some measurements in the Indiana showing those runoff carbon is eligible. But in other ecosystem, they might be. So that's one note. So based on all these understandings, so our objectives is trying to develop 30-meter NEE maps across the Midwest using ethical variance data, remote sensing, and meteorological data. And then the second is quantify the NEE difference between cover cropped and non-cover cropped fields. So here I want to emphasize we only use the dominant season for this presentation. And because at that time, the harvest carbon is 0, so you can just using NEE to indicate the carbon change. However, we are going to integrate the yield map and to use the equation we used before to quantify the net carbon exchange. Okay. So the methodology, we're using like 57 towers across the Midwest. Actually, it's more than Midwest states. And so those flux tower sites, including croplands and also grassland sites. So because they are pretty similar to cover crop type of vegetation, so we just put them together so that we have more towers. And we also use harmonized land site and Sentinel-2 data and DayMat for the climate variables, and then we're using random forest model. So after we modeled the NEE, we actually we were really lucky to have this high resolution conservation transect survey data, which you see they are pretty intensively across the Indiana. So the ground survey data has, 50,000 points across the Indiana for 8 years, and they also have cover crop species. This is a real rich data set and rarely to have it. So we kind of calculate the relative carbon change using NEE minus NEE baseline and divide NEE baseline. So I think the baseline is always important when you're trying to evaluate any practices. So here because we have the rich data, so we did calculate NEE dynamic baseline. So we can accounting the year and also the cash crop and in each like 10-mile grade. Which is really great. So some of the results, so I'm going to show you the drivers of NEE maps across the Midwest. So this is the indices for that. So the validation R-square value is 0.71. I think for NEE mapping is pretty good already if we don't using AI stuff here. So generally the indices including like the vegetation proxies and also agriculture management proxies and some climate variables. Something is just different from maybe compared to other NEE upscaling is some like the agriculture unique characters that we can actually characterize by the you remotely sensed data. So here I'm only showing the Indiana dominant season NEE map. Because the Transect data is across the Indiana. And then here's the results for like different cover crop species, how much they can uptake the the carbon. So the different species, you can see they actually varies differently. So the y-axis is relative carbon change and x-axis for the different species. So the first column is the reference. So the stars means everything is a significant difference. So generally the different species so this is also for the maize fields, it's going to uptake like 2.4%. And then this is the same for the soybean fields, they're pretty identical, the figure. So also different species just have a different ability to uptake carbon. So this is a figure is really interesting. So the different, so I separate the species into different cover crop quantity, right? So especially for soybeans, you could see really significant trend that if you have more biodiversity in the cover crops in that field, you can take carbon more efficient. So the last figure, the last results is we separate into the adoption period. So for some sites, they only adopt for 1 year, but then the other sites, they might adopt him for more than 3 years. Again, for the soybean fields, if you adopt him for multi-year of cover cropping, you usually uptake carbon more. Again, this is just take-home messages, and also timing is gone. So the high-resolution NE mapping across the Midwest is feasible, and the cover cropping, actually, we can measurably enhance carbon sequestration, and when we can also indicate that. So great diversity and longer duration also further amplifying this effect. However, our results are likely represent the upper limit of the SOC change from the cover cropping because we're only using the dominant season right now. So the next step, we're actually going to integrate the yield data to link the NEE to the SOC change across the Midwest and for the whole year. Thank you so much.
Thanks, Jen. We got time for one question as we just transition into the panel. Cove.
Thank you. For the multiple cover crops and the relationship there, when they do the multiple cover crops, is it one at a time or in a sequence, or like one at a time, meaning one following another, or all at the same time?
They're all the same. So they have the data just the same. This field, they have this species. So you do know how many species they have in that field.
So all mixed together.
Yeah, mixed together. Yeah.
Thank you.